You eat right, you try to get your steps in, maybe take a handful of vitamins every morning, all with this universal hope of keeping the machine running smoothly as you age.
Right. We all want to stay healthy.
Exactly. But what if the biggest obstacle to your health wasn't like environmental stress or an external pathogen? What if, right now, as you were listening to this, there was a microscopic horde of zombies living inside your own tissue, actively working to tear the building down from the inside?
I mean, it sounds like a horror pitch.
It really does.
But from a biological standpoint, this is actually one of the most stubborn puzzles in modern medicine. The cellular aging process isn't just about things slowing down, it is about microscopic sabotage.
And that is exactly what we are getting into today. We have this fascinating article from Science Daily that came out this past May 15th, 2026, about a major aging breakthrough at the Mayo Clinic.
Yeah, it completely reframes how we look at this whole zombie cell problem.
It really does. So the mission for today's deep dive is to understand how a totally unexpected, arguably crazy idea from a casual water cooler chat might change the future of treating aging, cancer, and Alzheimer's disease. Okay, let's unpack this.
Well, the most profound discoveries in science almost always happen when we take a stubborn, decades-old problem and force ourselves to look at it through a completely unorthodox lens.
Right.
The field of aging research has been stuck on this specific hurdle for a long time, and it really took a jolt from an entirely different scientific discipline to break the logjam.
So before we get into the breakthrough, let's start with the villains of this story, right? These microscopic zombies. I mean, anyone who follows biology knows that cells divide, and eventually they undergo apoptosis.
Right, programmed cell death.
Exactly.
Yeah.
They clear out to make way for fresh cells. But we are talking about senescent cells today. They skip the dying part.
Yeah, they experience severe stress or DNA damage, and instead of undergoing that programmed apoptosis, they enter a state of permanent arrest. They stop dividing, but they absolutely refuse to die.
They just hang around.
They hang around, but the real threat isn't just that they take up space. It is what they actively do while they are in this arrested state. They develop something called a senescence-associated secretory phenotype, or SSP.
SSP, right. They basically start pumping out this toxic mix of chemicals.
Exactly. Inflammatory cytokines, chemokines, proteases. They are essentially spewing a highly reactive, toxic cocktail that breaks down the structural integrity of the surrounding tissue.
Wow, so they are actively doing damage.
Oh, severe damage. They cause chronic inflammation. And even worse, that toxic secretion can actually trigger neighboring, completely healthy cells to become senescent as well.
Oh, wow. So it's contagious.
It is a biological chain reaction. This accumulation of senescent cells is a primary driver of the physical deterioration we call aging, and it's heavily linked to cancer progression and neurodegenerative conditions, like Alzheimer's.
So the obvious answer seems to be, well, let's just go in, find the zombies, and clear them out. We just need to develop drugs that kill them.
Sure, and the field of senolytics drugs designed to clear senescent cells has been working on that for years. The massive, incredibly frustrating obstacle is identification.
Finding them.
Right. To remove them, you first have to find them. And senescent cells don't have a universal neon sign pointing them out. They are essentially hiding seamlessly among billions of totally healthy cells in living tissue.
They blend right in.
Completely. The biomarkers we currently use to identify them are notoriously unreliable.
It feels like having a terrible squatter in an apartment building. Like, they stop paying rent, they refuse to leave, and they start actively trashing the hallways and blasting music at 3 a.m.
That's a great way to look at it.
But the catch is, they wear the exact same clothes and look exactly like all the good tenants. So you want to evict them, but you literally can't tell who is who.
Exactly. And if you try to forcefully evict that squatter with a heavy-handed traditional medical intervention, well, the collateral damage to the rest of the apartment building is immense.
You end up hurting the good tenants.
You end up harming healthy cells. So you need a way to spot the squatter with absolute precision. Because these cells are so good at hiding, the traditional biological tools we use just weren't cutting it. We needed an entirely new approach to the hardware itself.
Which brings us to the serendipitous part of this whole story. Because a direct, traditional attack wasn't working, the solution came from a completely different scientific discipline.
It is a classic tale of cross-pollination.
Yeah, so we are at the Mayo Clinic, and two graduate students happened to cross paths at a scientific mixer. You have Keenan Pearson, who was studying brain cancer and neurodegenerative diseases under Dr. Jim Marr III.
Right. Completely different department.
Exactly. He was focused on a highly specific type of molecular screening technology. And then you have Sarah Jackham. She was completing her research in Dr. Nathan Labrazor's lab, and her entire focus was on aging and these senescent zombie cells.
Right.
So they start chatting, and Pearson wonders out loud if the technology he uses for brain cancer could somehow be adapted to solve Jackham's zombie cell hiding problem.
And what's funny is Pearson actually admits in the source material that he had no idea how to actually culture or prepare senescent cells for testing.
That wasn't his thing at all.
No, it was entirely outside his wheelhouse. But it was Joachim's exact expertise. They each held half of a map.
So they take this idea to their mentors, including researcher Darren Baker, whose work focuses heavily on therapies targeting these exact cells. And Dr. Marr, Pearson's mentor, actually calls the concept crazy when he first hears it.
Yeah, crazy but intriguing enough to investigate further is how he described it. And to their credit, the mentors fully embraced the risk. They loved the synergy of two completely different research areas.
Which is pretty rare, right?
Very. And once they got the green light, the early experiments produced such encouraging data that they realized they had to scale the project up immediately.
Right, they had to bring in additional graduate students, Brandon Wilbanks, Luis Prieto, and Caroline Doherty, just to handle specialized techniques like advanced microscopy and tissue analysis.
The whole project just snowballed.
It did. But, I have to push back a little on the broader scientific landscape here, because we pump billions of dollars into medical research globally. Why does it take a random serendipitous chat at a water cooler for these two halves of the map to find each other? I mean, are we really that siloed?
It is a fair question, and it raises a fundamental issue about how modern research is structured. Science has become hyper-specialized, often by necessity.
Because there's just so much to know.
Right. To push the boundaries of human knowledge today, you have to spend your entire career becoming the absolute world expert on one tiny protein pathway in one specific region of the brain. That deep dive is necessary, but it inherently creates blinders.
You lose the forest for the trees.
Exactly. You often have no idea what the lab 3 doors down is doing, let alone a completely different department.
It's like you get so focused on the chemical composition of the paint that you forget you are standing inside an art museum.
That's a perfect analogy. And the academic funding and publishing models, they actively encourage staying in that highly specialized lane. Grants are awarded for hyper-focused, incremental progress within established paradigms.
So no one is funding the crazy crossover ideas.
Not usually. That is why creating unstructured spaces for open dialogue where different disciplines can organically collide is so crucial. It prevents that hyper-specialization from blinding us to a tool that might already exist right down the hall.
Well, speaking of that tool, we know how the idea was born, but what exactly was the crazy tool they decided to use to find these squatters?
They utilize something called an aptamer.
An aptamer. Okay, so what makes an aptamer different from traditional genetic tools?
Well, aptamers are short strands of synthetic DNA or RNA, but we aren't using them for their genetic code the way we usually think about DNA holding the instructions for life.
Right, not like the blueprint.
Exactly. DNA is normally a double helix, right? Like a zipped up ladder. Every chemical base on one side is bonded to a partner on the other side. But when you synthesize just a single strand of DNA, those chemical bases are suddenly left without partners.
They get lonely.
Very lonely. They desperately want to bond with something.
So they start grabbing onto themselves.
Yes, they fold back, crumpling up into these highly specific, complex, three-dimensional shapes. You get hairpins, loops, and bulges. It is basically molecular origami.
Well, that's cool.
And because of these unique, stable 3D topographies, aptamers can frequently attach or bind to specific target molecules like proteins on the outer surfaces of cells. Think of an aptamer as a highly specific puzzle piece that only fits into one exact slot.
Okay, here's where it gets really interesting for me.
Yeah.
Because you don't actually need to know what the slot looks like beforehand.
Right, you don't.
I look at aptamers like an impossibly huge ring of microscopic skeleton keys. You have millions of these random keys, and you have a locked door, the zombie cell. You don't know the shape of the lock, so you just keep throwing different keys at the door until one of them turns the tumbler.
That is exactly it. But the scale of the keyring they used in this experiment defies logic. Working with mouse cells, the Mayo Clinic scientists didn't just screen a few thousand sequences. They screened over 100 trillion random DNA sequences.
Wait, hold on. 100 trillion. I mean, how is that even physically possible? You can't put 100 trillion different samples under a microscope, one by one, to see what sticks. That would take 1,000 lifetimes.
No, you definitely don't do it
So
visually.
how do you actually screen a number that large?
You do it chemically through an evolutionary process called CX. You synthesize a massive liquid library containing those 100 trillion random aptamer shapes. You take a culture of your target, in this case, the senescent mouse cells, and you basically flood them with this ocean of aptamers. And the vast, vast majority of those shapes won't fit anything. They just float there.
So then you watch the cells.
Yes, you literally wash the sample. Everything that didn't physically bind to a senescent cell gets washed away. Then you change the chemical conditions to detach the tiny fraction of aptamers that actually stuck.
The ones that fit the lock.
Right. You take those successful binders and you use an enzyme process like PCR to multiply them by the millions. So now you have a smaller pool of much better keys.
And then you just run it again.
You run it again. But this time you might add healthy cells to the mix to act as decoys. If an aptamer sticks to a healthy cell, you throw it out.
Oh, because you only want the ones that exclusively bind to the zombie cells.
Exactly. You repeat this chemical washing and amplifying cycle over and over until you have filtered 100 trillion random sequences down to a handful of highly specific, incredibly sticky keys.
Wow.
Out of that massive starting pool, they successfully identified several rare aptamers capable of binding exclusively to the senescent cells.
So the key finally clicked.
It did. And once those specific aptamers attached, they flagged the zombie cells. They essentially painted a giant, glowing target on the squatter's door so the researchers could clearly differentiate them from the healthy tissue.
That is amazing.
What's fascinating here is how perfectly this establishes a new paradigm. It proves that synthetic DNA technology can reliably distinguish sick senescent cells from healthy ones, even when traditional biological markers completely fail.
But here's the crazy part. Finding the cells was the primary goal, right? Just flagging them.
Yeah.
But the way the chemical wash process actually found them accidentally unlocked a completely separate biological mystery.
It did. And this is where the sheer elegance of the experimental design really shines. Dr. Ma pointed out something crucial to date. There aren't universal markers that characterize senescent cells.
Right. There is no single known feature identical across every zombie cell.
Exactly. Because of this, they specifically designed the aptamer screening to be entirely open-ended. They didn't pre-program the synthetic DNA to look for a specific known protein.
Wait, so we basically built a microscopic molecular bloodhound, took it off the leash in a forest of 100 trillion possibilities, and it ran off and found the target by sniffing out a scent that we humans didn't even know existed.
That is exactly what happened. When they analyzed the rare aptamers that successfully stuck to the zombie cells, they had to work backward. They investigated what exactly on the cell surface the aptamer was holding onto.
They had to find out what the lock was.
Right. And they discovered that several of the successful aptamers had attached to a specific variation of a protein called fibronectin.
Fibronectin. Okay, what normally does fibronectin do?
Well, fibronectin is a well-known extracellular matrix protein. It is basically the scaffolding outside of cells that helps them stick together, and it plays a huge role in tissue repair and wound healing.
Okay.
It is very common on the surface of mouse cells. But this specific variant of fibronectin, the one acting as an exclusive beacon for senescence, was a brand new puzzle piece.
So we didn't even know this variant was a red flag before this.
No, we didn't. And the researchers openly admit they do not fully understand the mechanism yet. They don't know why the cell alters its fibronectin scaffolding when it becomes senescent.
But I guess that almost doesn't matter right now.
It doesn't. They have a massive, unexpected clue that will help the scientific community investigate the fundamental biology of what makes these cells unique in the first place. It is inductive reasoning at its finest. You observe the outcome, let the biology tell you what is important, and then work backward to understand the cause rather than forcing a rigid hypothesis.
Identifying this fibronectin variant in mice is undeniably a massive step for fundamental biology. I want to ground this for the listener, though.
Sure.
Why should someone, walking their dog or commuting to work, care about a mouse protein today? Like, what does this actually mean for the future of human medicine?
Well, the standard necessary scientific caution definitely applies here. This is an early step involving mouse models and cell cultures. The researchers emphasize that significant, rigorous studies are required before we can guarantee that aptamers will reliably identify senescent cells in humans.
Yeah, because a human is a lot more complicated than a petri dish.
Infinitely more complex. Moving from an isolated lab dish to a living human patient is a massive leap.
So nobody is getting an aptamer injection tomorrow.
No, definitely not tomorrow. But the theoretical advantages over our current technology, however, are staggering. Dr. Pearson notes that right now, the standard method for distinguishing different types of cells relies on antibodies.
Okay.
And antibodies are biological molecules. They require animals or complex cell cultures to produce, which makes them incredibly expensive. They can be unstable, and they are notoriously difficult to modify once you have them.
But aptamers are different.
Aptamers are synthetic DNA. We can literally manufacture them chemically in a machine. They are vastly less expensive, highly stable, and infinitely adaptable.
So what does this all mean? What is the ultimate end game if this technology translates to human patients?
Well, if we connect this to the bigger picture, this breakthrough has the potential to be much more than just a diagnostic tool. It is not just about taking a microscopic roll call and figuring out which cells are the bad ones.
Right.
Because these aptamers can perfectly and exclusively target the zombie cells, scientists believe they could eventually be engineered as delivery vehicles.
Like you attach a payload to them.
Exactly. You create an aptamer drug conjugate. You attach a therapeutic drug, perhaps a medicine specifically designed to force the cell into apoptosis directly to the synthetic DNA strand.
And then you just introduce it into the bloodstream.
Right. The aptamer travels through the body, completely ignoring billions of healthy cells. It finds the fibronectin variant on the zombie cell, clicks into the lock, and delivers the medicine point blank into the bad cell.
That changes everything about how we handle systemic disease. Because the current model of medicine for something like cancer or severe cellular dysfunction is often just a shotgun blast.
Yeah. Unfortunately, yes.
Systemic chemotherapy essentially nukes the whole area, and we just hope the bad cells die slightly faster than the healthy tissue. But this aptamer delivery system would be absolute sniper-like precision.
That is the hope. Highly targeted therapeutic approaches eliminate the collateral damage. You evict the squatter, and the rest of the apartment building doesn't even know you were there.
Man, what an incredible journey. To recap everything we've been over today, we started with a casual, seemingly crazy chat between Keenan Pearson and Sarah Jacob.
2 grad students at a mixer.
Right. And that cross-pollination of ideas gave birth to a project that used an evolutionary chemical wash to screen 100 trillion synthetic DNA keys, eventually finding the exact ones that fit the hidden locks on the microscopic zombies actively degrading our tissues. It is just a stunning piece of science that redefines how we hunt disease.
It really is a profound reminder that the answers to our most difficult medical problems often lie right at the intersection of entirely different disciplines. Knowledge becomes exponentially more powerful when diverse perspectives collide, and we really have to constantly challenge our underlying assumptions about what tools are allowed to be used on any given problem.
I love that. And I want to leave you, the listener, with a final provocative thought to chew on today. We have established that these senescent zombie cells are inherently tied to the physical aging process. They are the engines of chronic inflammation and tissue degradation.
They are.
So, if we now have the foundational blueprint for a microscopic smart bomb that can seek them out, identify them by their unique scaffolding, and eventually deliver point-blank treatments to clear them away, are we standing on the precipice of not just curing age-related diseases like Alzheimer's, but actually re-engineering the human lifespan?
It is a massive question.
If science can eventually go in and continuously, safely clear out the cellular garbage that makes our bodies break down, what does that mean for the natural human life cycle? It really makes you wonder what the absolute limit of the human machine actually is once those microscopic zombies are finally evicted for good.
It's something to think about.
Thank you so much for joining us on this deep dive. Keep asking questions, keep wondering about the world around you, and we will see you next time.